Hello dear learners, in this video we are going to talk about the transposable elements in eukaryotes. So here we have to discuss the three major cases. One is the transposable elements found in yeast, then we'll talk about the transposable elements found in maize, then we'll talk about the transposable elements which are found in Drosophila, and then additionally we are going to talk about the transposable elements found in humans. So in total four cases, but major ones are these three. Even we'll talk about the humans also. So let us talk about the transposable elements in eukaryotes. So we will directly come to the yeast because this is just the introductory paragraph which will be covered also here. So when we talk about the elements in yeast, these are called as Ty elements. The Ty stands for transposon yeast. So inside the genome of the yeast, there are certain elements which are the transposable elements, and since these are for yeast, they are commonly called as Ty, that is transposons for yeast. Now you have to remember this, that they are the family of retrotransposons, means their mode of replication will be by the transcription and reverse transcription. And please remember this, that the transposable elements in the yeast are very similar to that which are found in the retroviruses. So we'll talk about the similarity of this with those of retrovirus also. So you have to talk about, we will see this later on. Before that, you see that there are approximately 30 copies of Ty elements, up to 30 copies also, which are present here in one genome only. So one yeast cell contains one whole of the nuclear DNA, and that DNA can have up to 30 copies of the Ty element. Now inside the Ty element there are certain unique sequences which are called as the delta sequences. So here this pink colored ones are the delta sequence 1 and 2, light pink ones. These delta sequences are approximately 334 base pair long. They are very much analogous, or they are very much functionally similar to the long terminal repeats found in retrovirus. So when we talk about the genome of retrovirus, there also they have some of the repeats here, and these are called as long terminal repeats. So what this text is trying to say, that these delta sequences are very much analogous to these long terminal repeats. The delta sequences code for certain proteins, and these proteins are also very much similar to what these LTRs code for. These LTRs code for GAG and POL genes. These are the GAG and POL genes which are present in the retrovirus here, and these are very much similar to what the particular proteins are encoded by the uh, delta sequences of the yeast. Now these delta sequences have also one more very special feature. They contain promoters, and the promoters are very strong. They can translate this whole of the transposon. They can transcribe this whole of the transposon, not translate, transcribe, sorry, so that the RNA can be made. Also, these particular promoters found in the delta sequences are so strong that they are even capable of transcribing the other genes of yeast. Means they have dual function. One is that this promoter will transcribe this particular Ty element, and secondly, if it is present in the vicinity of a gene, not the Ty gene but any other gene, then what happens is, here this promoter is so strong that even it can do the transcription of that gene. So it is a very strong promoter. Then in between there are the genes present, which are Tya and Tyb. These genes code for certain enzymes and proteins like protease, integrase, reverse transcriptase, and RNase. So this is all about the transposable elements in yeast, which are very much similar to retrovirus. Hence they are called as retrotransposons. Moving on to the next, that is the transposable elements in maize or corn, and these are the AC and DS elements. Before talking about these elements, you should know that These elements were discovered by a scientist whose name was Barbara McClintock. So McClintock actually spent much of her long career studying all of the properties of AC and DS elements, and afterwards when the molecular techniques were discovered and were developed, using those molecular techniques actually these AC and DS elements and their effects on the coloration of the kernels of maize was studied, and later it was proved that whatever Barbara McClintock was saying, she was right. So basically AC and DS elements, they control the pigmentation, or they control the color in the kernels which are present in the maize. So generally these kernels are yellow in color, but we have seen, or even if you have not seen, you can see here, there are certain spots of it. Mostly the whole of the kernel is yellow in color, but then there are certain pigmented spots. These spots are also called as streaks. More prominently, these are called as sectors. This pigmentation is controlled by the AC and the DS elements. These are nothing but the transposable elements. So let us see their mechanism, that how this happens. You can see here that this is what the AC element looks like. The AC element consists of the transposase gene, and then it consists of the flanking direct repeats and inverted terminal repeats. Here are the DS elements: DS9, DS2D1, DS2D2 and DS6. All of them are very much similar to AC element, but there is one problem. All of them do not have a proper gene which can code for transposase enzyme. It means that there are certain deletions in the gene which codes for this transposase enzyme. So these DS elements themselves cannot be mobile. So as I was explaining in bacteria, what happened there? The insertion sequences on the either side of that complex transposon was helping, I can say complex transposon as this. So they were helping it to be cleaved. Similarly, the AC element which is present in the close vicinity of DS element will help it to cleave, and then this will move to the target sequence, and once it moves to the target sequence it will stop the pigmentation. That is the whole crux of the pigmentation. So here this transposase gene codes for the transposase enzyme, and this transposase enzyme works upon the particular sequences of the DS elements. So as I was talking about here, let us move into the details and let us try to see that how this happens. So normally if I say that the maize is there, and the kernel of maize, if we are talking about, the kernel of maize can have three kind of genotypes. We are assigning a gene which is capital C. This capital C, let us assume, codes for the pigmentation. So those kernels which have small c, they will never be pigmented. We know this. If they are capital C, then they will always be pigmented, and whole of that will be looking purplish in color. But if they are heterozygous, then the problem arises. So in that particular gene, what happens is that the AC element is present, the DS element is present, and then the C